as I was saying, depending on the nature of the genome of the virus, whether it's DNA, RNA, and whether it's single-stranded or double-stranded, will determine how the virus replicates. So the objective of this lecture is to talk about the so-called Baltimore classification, to talk about the strategies of replication, which is dependent on whether the virus is DNA or RNA. And this relates to the so-called Baltimore classification. And then to talk about the problems we have of naming viruses. So before I start with the Baltimore classification, let's just talk about this COVID-19. The press calls it COVID-19. Everyone knows it as COVID-19. But in actual fact, COVID-19 refers to the disease, not to the actual virus. The real accurate name for this virus is SARS-CoV-2. SARS, because the first SARS virus that was discovered was in 2003 in Mexico, which stands for severe acute respiratory syndrome. So this just goes to show how problematic the field of virology is in naming viruses, because some viruses are named depending on the type of disease they cause, like hepatitis virus. Some viruses are named based on where they were discovered, like Ebola virus or Bunyamuera virus. So there is no real order or structure to virus nomenclature like you have with bacteria, which are named based on the Linnaean system. So because of this, then in the 60s, this virologist called David Baltimore, who was the one who discovered retroviruses, came up with this idea of grouping viruses based on their type of nucleic acid they contained, whether they were DNA viruses or RNA viruses, or whether they were double-stranded or single-stranded. Because if they had similarities in this nucleic acid, that gave them more relationships with other viruses having the similar genomes in terms of replication processes. Nowadays, where our sequencing has been a lot more sophisticated, we now have more accurate ways of grouping different viruses in terms of relationships to create these so-called family trees to determine genetic identity or relatedness to group these viruses. But nevertheless, the Baltimore classification you'll find in virology textbooks and is still one of the most practical ways of trying to understand this diverse kingdom of organisms. So before we understand the Baltimore classification, we need to understand what we call the central dogma of molecular biology, that within our eukaryotic cells, or all cells, that we start off with a double-stranded DNA template. And that double strand consists of what we call the plus strand and a complementary minus strand. Now, if we had to transcribe and translate a particular gene within this DNA, the information for that messenger RNA has to come from a minus. Huh? What? So from that minus DNA strand, you get what is called a plus strand messenger RNA, right? And so that from this messenger RNA, you can then translate your codons to give you your amino acid sequence, which makes up your protein. But then remember that if your virus is a single-stranded minus strand, to create more minus strand DNA molecules, you need to first make a complementary plus strand so that you can read off from that plus strand template more minus strand DNA. And vice versa. If your virus is a plus strand, you need to make a minus strand to make more plus strand. So you always need a double strand, whether it's DNA or RNA, in order to create more of those molecules. Now with, so this is, as just to repeat the central dogma, you have double-stranded DNA, you transcribe from this minus strand a plus strand messenger RNA, which you can then create protein. And from this, in this process of creating more double-stranded DNA, using this enzyme called DNA-dependent DNA polymerase. To transcribe to get messenger RNA, you are using a DNA-dependent RNA polymerase. To translate that messenger RNA into protein, you're using ribosomes, right? But now, so this is in a eukaryotic cell. But eukaryotic cells do not have RNA-dependent RNA polymerases in the case of an RNA virus infecting a cell. So, unlike a DNA virus, which can use eukaryotic enzyme, the DNA-dependent DNA polymerase, to replicate its own genome, if it's an RNA virus, it has to make this enzyme from scratch because the host cell it's infecting does not normally have this enzyme. Okay? So then, once the virus infects a cell and manages to make this polymerase, it can then recreate, if it's a positive strand RNA virus, remember, in order to make new copies of itself, you have to have double-stranded. So if this virus is a plus strand RNA, it first needs to make a minus strand RNA complementary strand that it can use as a template to make more plus strand copies of itself in order to be packaged into the new virus particles. So now we have this problem. If the virus is a plus strand RNA, okay, then it needs to have, now, remember that I said that messenger RNA, by convention, we call that plus strand because it's read off by the minus strand, all right? So if a virus is a plus strand, That means that that plus strand genome is essentially functionally messenger RNA. Okay? So that means that when this virus infects a cell and exposes its genome, which is a plus strand RNA, that plus strand RNA is essentially messenger RNA. So that the host cell that it's infecting can use those ribosomes to read off that genomic RNA to create virus proteins. And one of the first proteins that it makes is this enzyme, RNA-dependent RNA polymerase, to create more plus strand RNA. Well, first of all, to make this minus strand RNA template so that it can read off more plus strand RNA molecules. You all get that? That's a very important concept. But now, think about what if a virus is a minus strand genome. Now here, this minus strand genome is not a messenger RNA. So it means that when it infects a cell, the cell's ribosomes cannot read to make protein out of that minus strand. So in essence, what these viruses need to do is that they need a pre-made RNA-dependent RNA polymerase packaged within these virus particles. So they need a pre-made functional RNA polymerase enzyme carried with them in their virus particles, such that when they infect a cell, these enzymes can then create a plus strand complementary strand to the viral genome from which it can read off more minus strand genomes. So this is essentially the difference between whether the virus is a plus strand or a minus strand virus particle. The plus strand is essentially messenger RNA that can be read off straight away as soon as it infects a cell to create this polymerase enzyme, whereas the minus strand cannot use the cell to read off a new polymerase and has to have a pre-made one. You got a question? Then we come to this third type of RNA virus, which is in this case, in the case of HIV or even COVID-19, well, no, not COVID-19, it's not a retrovirus. In the case of HIV, these viruses have plus strand RNA. When they infect a cell, what they do is from that RNA template, they make DNA, minus strand DNA. So they have a hybrid between a plus strand RNA and a minus strand DNA. And from that minus strand DNA is then read a plus strand DNA to create a double stranded DNA. And that RNA molecule is degraded. Once you have this double stranded DNA, that can insert itself into the host cell chromosome. So you can get a latent virus. And this is brought about by this special enzyme called reverse transcriptase, because it's reverse transcribing. It's going backwards from the central dogma of DNA to RNA. It's going from RNA into DNA. So
time, is it in the human cells or is it in the viral?
It's only in retroviruses.
So they carry it with them, like the in their.
No, because remember, this is a plus strand RNA virus. So it's messenger RNA. So once it infects a cell, it can use the cell's ribosomes and transcriptase to make the messenger RNA to read off that protein. So it doesn't need to carry it with it.
So it's in their genome.
Yeah. So now don't worry about learning whether this virus belongs to group 1 in the Baltimore classification and this virus belongs to group 2 and whatever. This is just to show you what the Baltimore classification looks like. But be aware that of this virus kingdom, we can have viruses which are double-stranded DNA, viruses which are single-stranded DNA, viruses which are double-stranded RNA, viruses which are single-stranded RNA which are either of a positive strand, as in a messenger RNA, or a minus strand. We get these retroviruses which are single-stranded RNA which go through this DNA intermediate where they can go latent, and then this special group, the hepatitis B virus, which is essentially a double-stranded DNA, but then as it replicates it goes through a DNA RNA intermediate, but then back into a double-stranded DNA. So now again, don't worry, you will not be questioned to give me the details of the whole sequence of events of each group within the classification. But this is just to show you the progress or the process of replication of these different types of viruses. But what you do need to know is within each of these groups, I give you an example of these viruses, and these are common viruses. So herpes viruses you should know about because you will learn and hear a lot more about them next year. So within the herpes virus, so the main thing to know about herpes viruses is that they are DNA viruses and they are enveloped, and that most of them can undergo latency. In other words, they can shut themselves down off from the immune system and then be reactivated at a later stage. So because herpes simplex is transmitted by kissing, this is the one that causes, there's two types. There's herpes simplex 1, which causes the cold sores I was talking about. Herpes 2, which causes genital warts, which is caused by sexual contact. So these DNA viruses behave like a eukaryotic cell, where it's a double-stranded DNA virus. So it undergoes replication to create more double-stranded DNA, just like a cell does, using the same enzymes, polymerase enzymes. When you make virus protein, you get the DNA-dependent RNA polymerase making messenger RNA. From that minus strand DNA template, that gets read into protein. The new double-stranded DNA molecules then get assembled to make new progeny viruses. Very straightforward. For single-stranded viruses, and the example is this virus, parvovirus, which can cause fetal abnormalities in pregnant mothers. And this is essentially a single-stranded positive or a minus strand. So remember, to create more strands from a single strand, you need to create the complementary strand. So with this, from this single strand within this virus particle, you have this complementary strand to create double-stranded, which you can then read off to make messenger RNA and to make more single-stranded DNA. From the messenger RNA, you make more protein, which can then assemble into capsids to encapsulate the new DNA, single-stranded DNA molecules. Now we go on to the RNA viruses. And as I said, because these are RNA-dependent RNA polymerase, which are absent in eukaryotic cells, the virus has to make this from scratch, or it needs to carry a pre-packaged enzyme if it's a minus strand RNA. Now, one feature of RNA viruses is that there are many more mutations during replication of RNA viruses compared to DNA viruses. One of the reasons for this is that this enzyme, RNA-dependent RNA polymerase, has no proofreading capabilities. When you get replication of DNA, the DNA-dependent DNA polymerase has various enzymatic functions where it reads these nucleotides inserted into the newly made DNA template. If incorrect nucleotides are inserted, it can edit that transcript, cut out those incorrectly inserted nucleotides, and put in the right one. If it still can't get that right, it will induce the cell to undergo apoptosis. So it prevents mutations from happening. RNA polymerase does not have this ability. So it means that every time you get new RNAs being made with this enzyme, you can get more and more mistakes or mutated, altered gene base sequences inserted into the new molecules. So it's been calculated that generally with RNA viruses, you get an error rate of 1 in 1,000 to 1 in 10,000. Every 1,000 new RNA molecules, bases in this RNA sequence, you're getting one mistake, one altered base sequence. So with each replication cycle of new progeny viruses, you get accumulating more and more mutations, such that after 10, 50, 100 generations, there are so many mutations in these new viruses that they are quite different. Let's say there's 20, 30% difference between the new progeny compared to the original parental strain which infected the cell. This can have implications in terms of the immune response. So this is one reason why we haven't been able to develop a good or an efficient vaccine against HIV, the virus that causes AIDS. Because even though you can develop a good antibody response against the initial strain that is the vaccine strand. As the virus replicates, you can get mutations so that they are no longer the antigen is no longer recognized by the antibody that the vaccine creates, and so these break out. And similarly, with even though we can cure
حضارات قاعد بسخر يا زلمه مفيش حدا قاعد بيسخر
بالعكس انتم ملفات
even though we can cure
طب ايش فينا
HIV
نفر طيب؟
شو
or halt its spread to a certain extent with antiretroviral drugs, it's still problematic that with these mutations, that many of these people who are treated, the virus can break out and escape from the antiretrovirals. Again, because of this high mutation rate. So, because of this high mutation ability of these RNA viruses, this is particularly in the case of HIV, hepatitis C virus, and this current COVID-19, where we've been sequencing cycles of new generation of viruses, in particular epidemic blocks, and coming up and seeing new species of viruses with each replication cycle. These are called quasi-species. So now, with regard to the RNA viruses, we can either have double-stranded RNA or single-stranded. The double-stranded RNA is not much of a problem to look at, because here you have a minus strand and a plus strand, and so again, from that minus strand template, you make your messenger RNA, which can then make proteins and also create the RNA-dependent RNA polymerase to make more RNA and DNA molecules. And these can assemble into the new progeny virus particles. The common example of this type of virus are reoviruses, such as the rotavirus, which is a particular problem in daycare centers, where it causes diarrhea and vomiting in young babies. And this is spread through the fecal oral route. Now, if we go to single-stranded viruses, which are positive strand, and an example of this is the virus that causes polio, hepatitis A, and also the virus that causes the common cold. So this is one of the largest group of RNA viruses. And remember I said that if it's positive strand, it is essentially messenger RNA. So once the virus infects a cell, that positive strand can get translated into a protein. In this case, the first protein being this RNA-dependent RNA polymerase. This enzyme will then create more minus strands to read off more plus strand genomic RNA molecules. In addition, that messenger RNA can be read out to create more protein, and so you have more progeny in this replication process. Now, just based on the immunology side, just bear in mind that because this is a completely artificial from a eukaryotic cell point of view, eukaryotic cells do not contain double-stranded RNA. So when a cell sees double-stranded RNA within the cytoplasm, it gets recognized by NOD-like receptors, Toll-like receptors, and this will activate the innate immune response. And this is where you get stimulation of NF-kappa B, inflammatory response, interferons being made to degrade and kill these viruses. So these are the internal sensing of the innate immune system for these novel virus components. And so just to repeat, it's pretty simple for a plus strand RNA virus to have that red as a messenger RNA. Minus strand RNA viruses will not be red, so they need to carry their own RNA-dependent RNA polymerase enzyme with them. An example of this are these viruses which cause influenza, which are called orthomyxoviruses. So basically, once the virus infects a cell, that virus-specific enzyme creates plus-strand RNA, which acts as a template to make more messenger RNA. One thing to note, well, I already talked about the fact that with the influenza virus, the genome is segmented. So let's say that you have two strains, A and B, infecting the same host. So you will have 8 segments, say strain A having segment 1, 2, 3, 4, 5, and the other one, strain B, having 1, 2, 3, 4, 5. As these replicate, you can have mixing of, say, the first 4 segments from strain A mixing with the last 4 segments of strain B. So you get these hybrids of A and B strains coming out. What this creates is what we call genetic drift. Basically, this is in the case of avian influenza, where we're getting this recombination of 2 different strains to create a very different strain. And this is what the WHO is very worried with, because when you get something like this coming out, it means your immune system's never seen this before. And so these recombinant strains, through this process of genetic shift, can create, has the potential to create pandemics with high mortality. The year-to-year variations you get with seasonal flu due to the mutations through this lack of proofreading with RNA replication, that is called genetic drift. So you get genetic shift, these major earthquake changes due to this recombination, and genetic drift due to these minor mutational variations. And this is why for the flu vaccine, you need to get a new vaccine every year, because each year the WHO predicts that there might be, or there will be, a different strain hitting the region or the world compared to what you were vaccinated against the previous year. So now we have these retroviruses where the virus starts off as a single-stranded RNA virus. Through this enzyme, the reverse transcriptase, it makes an RNA DNA intermediate. The RNA is degraded so that from that minus strand DNA, you create a plus strand DNA. So you have a double-stranded DNA now. And this is called a provirus because this provirus can then integrate into the genome and then be part of the genetic information of the cell, switch itself off so you don't induce an immune response, and later on the virus can kickstart itself and start replicating again. And then these are the hepatitis viruses, which start off as a double-stranded DNA, partially double-stranded, circular. So it has a plus strand and a minus strand. When it infects, that plus strand is closed off. So it has more nucleotides to create a complete circle. So now this acts like a plasmid. And this is one of the reasons why it is almost impossible to get rid of this virus when you're infected, because this plasmid is very, very stable. There's no way, if a cell contains this viral plasmid, there's no way we can get rid of it. From this, you can read off messenger RNA and then create from here an RNA, which is then read through reverse transcriptases like retroviruses into DNA, but then that RNA is cleaved to end up with this double-stranded DNA in new particles. And finally, we have a group of viruses which are so small that they only have enough genetic information to code for one or two proteins, namely capsid proteins. So basically what they do is they have to piggyback with other viruses and use the genetic information of these other viruses to code for particular enzymes. And so they are called satellite viruses. And that means that these viruses cannot exist without co-infecting with one of their helper viruses. So in this case, you have adeno-associated virus. So this virus cannot, has to infect a cell which is already infected with another virus called an adenovirus. The adenovirus providing the enzymes and other accessory proteins for this small virus to function. Hepatitis delta virus is another virus which can only infect hepatocytes if these hepatocytes already have in them hepatitis B virus. Hepatitis B providing the necessary proteins for this virus to survive. In this case, any patients who are infected with both hepatitis D and B have a much poorer prognosis. They have a much higher chance of developing cancer, liver cancer. I've talked about these. And I've talked about these. So, as I said, because of genetic sequencing, we can now more accurately classify or give some structure on classifying viruses within these different families. But the thing is that this is so dry that it really hasn't taken off. So unlike bacteria, where we talk about Clostridium tetani in terms of species and genera, E. coli, and so on, even though we have this structure for viruses, amongst the general population and even amongst medical doctors, this hasn't really been adopted. The scheme hasn't really been adopted. Only amongst pure virologists who are more interested in this sort of stuff. So, as an example, again, don't bother about learning this, but one of the most common viruses is Epstein-Barr virus, right? Epstein-Barr virus is a herpes virus. It's part of the herpes virus family. It's called Epstein-Barr virus because it was discovered by this virologist, Joseph Epstein, and his research assistant, Elizabeth Barr, where he graciously used her name to name this virus. This was discovered in the late 50s. This virus causes B-cell lymphomas, also Burkitt's lymphoma. So then in terms of this dry classification, this virus is a member of the herpes virus family, and it's within this, it's clustered into these different names. But again, don't worry about this. This is just be aware that this type of nomenclature is around. But for your purposes, you will still think of this as Epstein-Barr virus. You will still be taught that this is Epstein-Barr virus next year. This is an example of what happens with genetic sequence. So this is the most recent Ebola virus outbreak. You have your initial strain infecting a community. Over a couple of weeks, you start sequencing isolated strains from people, and you start getting all of these minor variants. And then an infected person might move to another town, infect another population, you infect. And so over a period of 6 months to a year, you can get from this initial parental strain all of these other types of strains coming out. And this is what we mean by quasi-species. And this shows the amount of variation or mutability we get through the course of an infection epidemic or a pandemic. And the same thing is happening now with the COVID-19 virus. Okay, that's it. So again,